Okay. So hi, welcome from me as well. Good to have you back. As we didn't finish the slides for the last session, so the introduction in MRI basics, we just go through the functional MRI basics and quickly through the kind of standard analysis pipelines for the different MRI modalities. But I'll try to make it quick because we have the topic for this week's pending as well. So yeah, let's see how this goes. Yeah, we finished up last week with the basics of diffusion-related imaging, and now we are at the stage of the functional MRI. And yeah, just for a quick overview of the measurement principles, because it's a quite indirect measure of brain activity. The cool thing is it's non-invasive, but one gotta make some assumptions about what happens in the brain and what we can measure. So it's quite indirect. So the underlying mechanisms behind that, so the biological mechanism is that an increase in neural activity leads to an increase in cerebral blood flow and blood volume, because activity needs energy, and so the energy supplies is increased, but it's kind of an overshoot. So you can measure the metabolic rate of oxygen consumption based on neuronal activity, and there's even more oxygenated blood, so more oxygen supplied for the activity. So there's an overcompensation. That means there's more oxygenated blood than it is metabolized. And this is a cool feature because there are different magnetic properties of blood which is oxygenated in comparison to a deoxygenated hemoglobin in the blood, because the deoxygenated hemoglobin, so the blood that has been metabolized, disturbs the local magnetic fields, and there are more inhomogeneities, and this affects the MR signal, and you can see a reduction. So desoxygenated hemoglobin leads to a reduction in the MR signal, and the cool thing is that you can measure this overcompensation with oxygenated blood, and so we have a stronger signal, which can be from which then neuronal activity can be inferred. Yeah, this is quite complex, but stronger signal in MR can be linked to an increase in neural activity. This is quite indirect because we have blood flow and that we can measure, and this signal can then be modeled with the hemodynamic response function. So it's kind of a chain of responses from a stimulus through to a neuronal activity, and then finally to a measured signal. And this is all governed strongly by the blood's response to the signal, which can be characterized by this function, the hemodynamic response function. So there is an initialized stimulus at time point 0, and the bold signal corresponding to this activity is then rising in time, and there's an overshoot and a small undershoot, and this is in theory a model of how the blood flow reacts to a stimulus. So the measurement is, or this function would be expected in a perfect noise-free scanner when the subject receives a really short and sharp stimulus. So you see, there are quite some assumptions you gotta make to measure this neuronal activity with MRI. But basically, it's a sequence of MRI scans, which is characterized by repetition time, TR. And so you have for each voxel one measure for each time point in defined by the repetition time. And with that, you can then interpolate a time series for each voxel over time for the whole brain volume. And there you have it's indirect, but you have a quite good measure of brain activity for the whole three-dimensional brain in time. Here you can see how this looks like by bare eye, the different images in time. So you barely see anything, so they need some preprocessing steps to make the signal visible. And then this looks like this. Here in the middle, you get a time series for each voxel. If you place a subject in a scanner without asking her to do anything, you can compute a so-called resting state connectivity. So it's kind of a functional measure of how the different brain areas are correlating with each other. So you're measuring a signal, an average signal across one brain area, and you compute a correlation matrix between all areas. Sorry. And this is called resting state connectivity. Out of this resting state connectivity, you can then build some compartments that have specific characteristics to define functional networks that are closely correlating with each other. And so there you can infer that there is a functional relationship between those areas, which can be defined with these 7 networks, for example, by you and colleagues, where you can differentiate between different characteristics or different modalities of those functional networks. For example, the visual network, the dorsal attention network, or ventral attention network. So you can can build compartments out of it. Another thing is, which is a bit more complex, the HCP's multimodal cortical parcellation, which I'll refer to in the second talk, so the second lecture of today. So we'll skip this for now. So as a quick summary, the fMRI can be seen as a surrogate measure of neuronal activity. And the measure is the blood oxygen level dependent effect, so the BOLD measure. The important thing is that deoxygenated hemoglobin, so blood disturbs, or substrate in the blood disturbs a local B0, so the local magnetic field, which changes then the relaxation times and the MR signal that can be measured in the scanner. So ecoplanar imaging acquisitions are required for this kind of fast imaging for fast repetition time, and so-called gradient AQ sequencing are really sensitive to this T2 star contrast. I hope you remember some of those terms from the last week. Then we have a resting state fMRI, which provides information about the functional connectivity between certain brain regions. Okay, that's for the introduction, for the modalities. And now we can go through the basic or most common analysis pipelines for MR processing. And yeah, there I will go through the structural pipeline, the diffusion pipeline, and the functional pipeline, because everything what I'm telling you is also relating to the second lecture. Yeah, so for the structural pipeline, there are different steps applied. So the first one is intensity normalization. The second one is a brain extraction, because if you lay a subject in the scanner, you will also measure the whole scalp. And yeah, we are also only interested in the brain for this case. So for analysis, it's always convenient to extract only those voxels in the volume which we are interested in. The third step is the spatial normalization or registration for some group analyses, for example, segmentation and parcellation. So grouping different areas based on certain assumptions. Then cortical surface modeling. Why this is important, I will also go into detail in the next lecture. And then we can analyze the structural images, for example, with voxel-based morphometry. So we can measure how many gray matter voxels are in the image to build a model of the different tissue types in an individual subject. So for the first part, the intensity normalization, it's also called bias field correction. And yeah, the bias field is kind of a artifact which is resulting from kind of inhomogeneities of the magnetic field applied in the scanner. So there are variations in the sensitivity of the signal, and the human body is interacting with this magnetic field that is applied in the scanner. So if we get then an image, this can be distorted by this inhomogeneity of the initial magnetic field. The bias field is dependent on the strength of like 1.5 Tesla scanner. It really can affect the analysis. In a higher magnetic field, the biases are even higher. So in the 7 Tesla scanner, you definitely have to generate those called field maps, which help you for this bias field correction. So commonly used tools are the ANTS bias correction or the FSL suite. How this correction can impact your analysis, you can see here pretty well. So by bare eye, it's quite hard to see that here is an artifact present. But if you take a look at the histograms and the intensities, then you see in the first picture, it's quite hard to disentangle the different tissue types and their intensity strengths, because the bias field is dependent on the physical shape of the body as well. So certain areas are more prone to those biases, and this can disturb the whole distribution of signals. And if you apply this correction, then you can, by bare eye, differentiate between the different tissue types and their signal strengths. So here is the intensity histograms plotted. And in the segmentation, it's also important because if you're interested in how in the cortical gray matter volume, for example, you have to segment the initial MRI picture image into tissue types. And if you can't disentangle the tissue types, then you can bias, or this would lead to a bias in segmentation between those tissue types, and would inflate the amount of gray matter that is detected here, for example. Then brain extraction, the next step. It's the basic purpose, as I told you, to strip the skull and to focus only on the brain. Therefore, we apply a mask that identifies the borders of the brain and to extract only the voxels which comprise gray matter, white matter, or cerebral spinal fluid of those cortical and subcortical structures. And ideally, we want to also take a look at the brainstem and the cerebellum. So we have to carve out those structures as well, which are a bit asymmetrically appending to the cortex. Yeah. Then with applying this mask, we are not deleting the voxels that are not brain specific, but we're just saying that those are irrelevant. And those are, for example, the scalp, fat, skin, muscles, eyes, bones, and we classify then those as non-brain. Yeah, this reduces the risk of registration failure because the physical shape of the head is even more unique than the shape of the brain in this case, and yeah, the whole analysis pipeline is getting more robust because your algorithms don't have to care about structures we're not interested in. For example, here one quite good brain extraction procedure and one fail. There in the right side, you can see that the top of the brain is stripped as well. And on the other hand, you clearly see that the neck is and the lower part of the head is still present. There are different algorithms that use this brain extraction or that apply brain extractions, and you can set the parameters differently to fit to each single subject or to supply the right parameters for extraction for each subject. There's a difference between this brain extraction or skull stripping and cortical surface modeling. As you clearly can see here, this is a 3D render of a brain mask. And there you can only see the brain volume and the voxels, but you have no information about the cortical folding, so the differentiation between sulci and gyri, and this can be done then with another step at the end of the analysis pipeline, which is called cortical surface modeling, which we will come across later in the stage as well. Okay, as a little summary, brain extraction is really useful, but it's not essential. So analyses will work without this extraction process, but it removes the influence of those non-brain structures and therefore improves the robustness of operations. As we could see, it's not aimed to model the detail of the cortical folds. And it's just to apply a mask to define as one for brain and 0 non-brain voxels. As you can see, algorithms sometimes fail, as therefore a visual inspection of the results is always a good option. Okay, then spatial normalization and registration. What does this mean? It means that there are different coordinate systems, as we have seen with this affine transformation last week as well. So there are world coordinates, north or south, for example. There are anatomical coordinates, so the position in your body. And then there are image coordinates, like the position of each voxel in a 3-dimensional image space. And we have to somehow interlink those coordinates, which can be isolated or stand in isolation as well. And to bring like the body coordinates, which are differently in each subject, into a one consistent coordinate framework, there has to be some spatial normalization applied. So we need a common framework in which we can, for example, say that this voxel is always lying in the hippocampus. So we have to somehow transfer the knowledge we gain from individual subject, and we build a group average, but we have to somehow communicate where our findings took place. So where in the brain we find some exciting new results. So the anatomical space is then a coordinate system that consists of those 3 planes. So describe the standard anatomical position of a human. So the axial plane or horizontal plane is parallel to the ground and separates the head superior from the inferior. Then there's a coronal plane, which is like kind of perpendicular to the ground and separates the front from the back. And then there's the sagittal plane that separates the left from the right. To convey those front, back, left, right coordinates into an image space, there's one stereotaxic space. It's a coordinate framework which has decided to be a reference frame, which is based on anatomical landmarks. But it's kind of a human decision to say, okay, our 0 point is lying next to the anterior commissure. But this is a definition to have a common coordinate reference framework. And this stereotaxic space was originally used to guide like neurosurgery, but has to be proven to be a good template to be a representative average of a study cohort. So if you want to communicate results, then you can rely to the stereotaxic space. And if you report a coordinate in that space, other researchers know where in the brain this coordinate sits. Yeah, that's a good reason to maintain comparability across studies, across subjects, and to make imaging processing pipelines publicly available as templates, for example. So if you have then this coordinate framework and a group average, this is then can be then published as a template, which serves then as a common reference space or brain in the common reference coordinate framework. In order to spatially normalize brains, we have to align the images from one coordinate space to another. So from the subject coordinate space to the stereotaxic space, for example. And this is also called intra-subject alignment, so kind of co-registration. We are aligning the individual brain to a standard template, for example. And you can see how this is applied. You have a source image and a target image, and we have to transform the source image to fit the target image. Another image registration is then the inter-subject alignment. Oh, I think I mixed this up. So a co-registration is the intra-subject alignment. So you have two different modalities of scans, and you want to align the scans from one subject. And then there is the inter-subject alignment, where we have to align the subject brain towards the template space. So there are two different alignment types, which I mixed up right now. So, but basically it's the same process, but on the one hand, it's called co-registration because we apply multiple image sequences to the same subject, and we want to bring all to the same space, which is the subject coordinate framework. And the next step is then the image registration to compare the individual subject brain with others or with a group template. And that's called registration. Yeah, we apply, if this is a brain from one subject and this is the group average, then we have to transform the image to fit the group average. So we have all brain areas kind of aligned in the same fashion to report common knowledge or report your research findings across a group of subjects. There we have to be aware of a so-called group registration drift, which is kind of interesting because if you average or register many individual brains to a group template, for example, the MRI template, then the brain gets kind of inflated, so the brain volume increases. And yeah, there are ways to account for this, but it's always good to be aware that those processes can happen. So you can see here in the GIF at the top right corner of the slide, a kind of a slight registration between the subject space and group average MRI template. Those registration can be done in a volume or on a surface space level, which we come to in the second lecture as well. This registration is not trivial. There's a linear registration method and a nonlinear registration method, and there are many rotations and translations one can do with a voxel to shift it towards the group average. So for a use case, we have a subject space and we have to align, so co-registrate the individual modalities. But if we want to compare the subjects, we have to register them to a common coordinate framework, which is in this case a reference space, and this is called spatial normalization. And this offers then a way to map and compare the anatomy of individual brains across modalities and even across studies. So we have, if results are published in MRI space, for example, then other researchers can compare this result then with their own findings if they are also registered to the same template space. Okay, as a summary for the registration, it's a fundamental tool in MRI. The parameters for spatial transformations need to be specified, so this is quite a complex process, and which I haven't said yet, but the spatial transformation and the resampling requires the interpolation of intensity data. As our MRI intensities have no real unit, it is not as crucial, but you have to be always aware of the fact that each transformation and resampling degrades the underlying data. So if you're not interested in a group comparison, there's no need to initially register your individual brain to another one. So if you have an intra-subject comparison in a longitudinal study, it's also sufficient to co-register all the modalities of one subject, but to stay in the subject coordinate space, because then the data is not degraded as much as if you then apply the spatial normalization to a group average template. And as always, the results of a registration should be checked visually, because others are complex algorithms and things can go wrong. Okay, then a quick introduction to segmentation and parcellation, because this will also be part of the next lecture. And if I gotta keep track of the time, because otherwise the next lecture I'm always teasing is not gonna happen. Therefore, segmentation of brain tissues. Just a quick thing, because we talked about this, we can divide the tissue types into gray and white matter and cerebrospinal fluid. And yeah, each class can then inform the different aspects of the brain. And it's also useful to segment the brain in these components for further processing, because as you might know, that the cell bodies mostly lie in the gray matter of the brain, and the white matter mostly are consisting of the myelinated fiber tracts, so the connections between the neurons. And next to the segmentation into tissue classes, you can also segment the brain into different compartments, which we be focusing on the part 2. Then, as I said, the cortical surface extraction or surface modeling is then a more sophisticated step to reconstruct the cortical folding of the cortical surface, as you can see here. So you take out the three-dimensional brain volume and you construct a two-dimensional sphere out of it. So the outer layer, basically the surface, and there you can reconstruct in detail the cortical folding patterns. And the surface data, in contrast to the voxel volume data, is consisting out of a vertex mesh. So it's a two-dimensional mesh that is made out of vertices and that are made out of edges and the faces, so the area between those. And yeah, the most commonly used surfaces are the pile gray matter surface or the underlying white matter surface. So it's the outer boundary of the brain, and the underlying white matter surface is the boundary of the brain without the cortical ribbon. Why surface-based analyses? We will talk about this later as well. But to say it quickly, there's a dimensionality reduction from 3 dimensions into 2 dimensions. It increases the specificity of cortical activation patterns and connectivity results. And it's a way simpler registration because you don't have to think about how to shift and rotate voxels in 3 dimensions. You don't just have to think about how to shift the vertices in 2 dimensions. So as a summary, the cortical surface modeling is fitting surface models to inner and outer cortical gray matter surfaces, and therefore we need a good quality structural images. But if we have this reconstruction of white matter boundaries and gray matter boundaries, we can calculate the cortical thickness of those surfaces. Just because we have a 2-dimensional sphere, we can inflate or even flatten the surface for viewing purposes. So we don't have a 3rd dimension anymore. And if you inflate the brain totally into a ball, then it's called a sphere. And those versions of the surface can be generated and are really useful for surface-based registration as is done in FreeSurfer, for example. Yeah, and then there are several other advantages of surface-based analysis, and it can be also combined with volumetric analyses of those subcortical structures, which are in the middle of the brain and therefore can't be extracted with this cortical surface reconstruction. So as you've seen, I always said that each algorithm should be visually checked afterwards because there's so many things can go wrong with those pre-processing. And if we have artifacts in those processing steps, so each step has its own pitfalls, and we are applying many steps in a row. So we need some kind of quality control because things can go wrong at each steps, and especially with automated image processing pipelines, we have to take a look afterwards at the results for visual quality assessments. But there are, in fact, also automated quality control tools which extract kind of quality metrics from structural and functional MRI data, such as the signal to noise ratio or contrast to noise ratio, and can therefore compute kind of continuous measures of image processing quality. But it's always good to take a look at the brain in the end, because as you can see with the brain extraction, then the computer doesn't know that the lower neck is not part of the brain, but you have to define this. So, okay, I think I will skip this summary because it's saying the same things I was telling you the last 10 minutes. And we come into the diffusion MRI pipeline. And just quickly go through those because the pre-processing is kind of similar to the processing of the structural MRI data with some specific with a specific focus of to some things. After pre-processing of the data, we have to apply a model for the local fiber orientation reconstruction, and then we can build a model of a tractogram. So the white matter tracts from one gray matter voxel to another one. So there are several steps comprised in pre-processing of DMRI, but this is depending on how the data is acquired, and there's not for every step there's not all the time consensus. So we will just focus on the things that are consent in the community. So brain masking is always important. Then we have to apply some corrections for diffusion weighted specific artifacts, and then there is an image registration co-registration to the T1 weighted image, so to the structural scan of the individual. After pre-processing, then we have to reconstruct those local fiber orientations. And yeah, there are many choices of models which all influence the results in their specific way. And the methods vary in terms of the required data. So it depends on which data you have acquired. And yeah, there are a few factors that influence the choice for a given reconstruction method. So the number of shells, the acquisition sampling schema, so how the MRI parameters were set, and yeah, the available time to reconstruct the data, because each algorithm takes, and yeah, regarding to its complexity, can take a long time. And if you want to process hundreds of subjects, this might be yeah, at least something to consider. Okay. So for the tractography, as I said, then we try to reconstruct the fibers that go from one cortical area to another one. Then here's deterministic tractography, which estimates the fiber orientation and follows along these orientations to propagate those streamlines. So we need a method for getting the local orientation directions from a diffusion-weighted MRI datasets. So, for example, with a diffusion tensor model, then we need a set of seeds which to begin the tracking. So, from which voxel we want to begin, and then we want to try. We want to have a method for identify when to stop the tracking. So you can start on one gray matter voxel on the cortical surface, for example, and then track down the white matter tract you are reconstructing, but you have to define where to stop. So you can define, for example, to stop at another point of the cortical surface, and then you have the connection from one voxel of the surface to another one. So as a summary, there are a number of different processing steps. The combined correction of distortions is important because there could be also an interaction of different artifacts. The diffusion tensor models, for example, fit the data as a surrogate measure of the white matter microstructure. So the diffusion tensor imaging is kind of a simple approximation of the underlying diffusion signal. The tractography is used then to trace out the long-range anatomical connections through the white matter tracts. It can be applied differently. And out of this tractography, connectivity from this can be used to create connectivity-based segmentations of regions, so a connectome. And you can say which regions are connected, which other areas of the brain. So for the fMRI pipeline, just quickly, we take raw fMRI, then we apply brain extraction. The special case for fMRI is that we are taking a sequence of images across time, and usually subjects in the scanner are not completely lying calm, so there is head motion between the images. And in order to correct that, we need to register each image in time to one reference image. So it's kind of a co-registration in time. And there's another distortion correction. At the end, we have to co-register this fMRI time series again to the structural T1 scan of the subject. And for group analyses, we can then transform it to the MNI template space and do some regression, for example, if we want to measure a contrast in a specific fMRI task. Okay, I think this is exactly the same pipeline again. So we have an anatomical preprocessing and the pipeline of functional preprocessing, which has to be done both in that case, because we need a structural scan which is in a higher resolution for the subject to align it with the brain activity patterns measured for with the fMRI scan. Okay, yeah, as I said, the subjects are moving, so this motion and distortion correction is really important in fMRI. And the importance is not only that we get a kind of an inflated brain with these movements, but we get also reduced sensitivity. So without a realignment, we can lose some significant activation patterns. But also there's proneness to reduced specificity. So we can also overinterpret some findings which are solely relying on misaligned image sequences. Okay, functional and diffusion imaging is based on a set of this kind of fast equiplanar imaging acquisitions. So, for example, functional imaging data is not only 3-dimensional, it's 4-dimensional, because we have another dimension of time. So we have to correct for motion between the images, and there could be geometric distortions which have to be corrected as well. The motion correction is often a rigid body registration between the volumes, and the amount of type of motions can be often quantified using those parameters deducted from those registration processes. And those field maps I was talking about, then can be used to estimate and correct for those geometric distortions and B0 field inhomogeneities. But this is important because for field maps, this is another modality of brain imaging. So you can't, there are options to calculate the field maps out of other image modalities like the T1 or T2 images, but for precise correction for geometric distortions, another image must be planned beforehand and acquired for each subject in each session. Yeah, this is important if one will plan a fMRI study or an MRI-based study, that those maps have to be acquired as well for each subject. Okay, then this is a software called fMRI Prep, which is a quite nice processing tool for functional MRI. But I think I'll skip this because this is basically the same thing in another slide again. Okay, so as a summary for the fMRI pipeline, we have artifact correction as the first step and compensation of artifacts. Then we can spatially smooth, but I haven't told you anything about that because we will focus this in the second lecture. Yeah, but if you have heard about it before, so the smoothing increases the signal to noise ratio but lowers the effective spatial resolution. Then there is temporal filtering, which is used to remove those low frequency drifts. So if you have a time series, you can filter this with kind of low pass filters, high pass filters. Then there's physiological noise that cannot be removed. But for example, heartbeat. But if you separately acquire physiological measures, then one can compensate for those artifacts. And there is intensity normalization between subjects, and this scales the data by single values so that we have a consistent overall mean across all brains and all points in time. Because the MRI signal there's no standard reference unit, so each individual can emit different signals basically. There are different kinds of fMRI. So there's task-based fMRI, and resting state fMRI, but we will cover this later as well. Okay, maybe we make like 5 minutes or 3 minutes break, and if there are any questions, then please type in the chat, and then we can do a little Q&A. Okay. I see there are no urgent questions right now, so I will try to set up the second presentation for today meanwhile, and maybe there will pop up a little question in the way. Okay, can you see the slides again? Yeah. Great, thank you. Okay, I think then we can start with the second lecture of today. So it's the wrong title, but it's about the processing standards of the Human Connectome project, and especially the parcellation of the multimodal parcellation of the Human Connectome project. It's quite a specific and really prominent and highly cited processing pipeline, and why we are focusing on so deeply on those processing pipelines. We've talked about this last week as well, but for those who weren't there, just a reminder, there's a great impact of which processing pipeline you choose on your data. So in this study by Lindquist and colleagues, they asked 70 different neuroimaging laboratories to analyze the same data set, and each of the 70 different labs produced a different result. So there were no two groups that exactly used the same analysis pipeline, which you can imagine is quite difficult feature. So on the one hand, it's cool that everybody is developing a different strategy. So hopefully with that data is getting generalizable, but most of the time the consideration behind the processing pipelines are not well reasoned and slowly chosen by the availability of the software, for example, and not in close consideration of the data and which processing is needed for that. And as you can see here, there are different degrees of spatial smoothing applied, different statistical models, and different thresholds which leads to completely different peak activity patterns over the brain. So this is really problematic, as you can imagine, because we have exactly the same data, but the kind of processing and statistical model will impact your results greatly. This is for one hypothesis in the study. The probability to report a positive hypothesis as yes depends on the processing standards. So they have found out for one hypothesis different clusters of labs which have applied a more liberal, in this case, or more conservative processing steps, which impacts the probability to report a positive hypothesis. And therefore, kind of standard and really high quality processing pipelines are needed that everybody can refer to, and to establish a kind of a common framework for processing and for analyzing data, to get a more glimpse of the underlying ground truth every lab wants to investigate. For sure, adaptations are possible and important, because each data is different, and therefore processing pipelines cannot be applied directly to each set of data. There is no process button, but at least a framework, or the fact that this processing affects your data immensely is really important to know. Therefore, there was the Human Connectome project, which developed a really complex set of processing pipelines. And yeah, this was original 5-year project in America, with the aim to completely map the neural, structural, and functional connectivity of the human brain. There was this was a multi-site, multimodal imaging study, which also acquired a huge amount of genetic, behavioral, and clinical data. For the first data set, 1,200 young adults were scanned with tons of gigabytes of data generated afterwards. So the original project was from the Washington University and Minnesota University in Oxford, this HCP consortium, which created this young adult set of images from subjects in the age between 22 and 35. Then there was a lifespan connectome project afterwards for young and developing and older subjects from 5 to 21 and 36 to over 100 years old. So the whole human developmental age range was covered in the end. Data is now stored in the Connectome coordination facility, because this project is finished, and now it focuses on data sharing and ongoing disease connectome project, for example, for psychotic disorders and other data requirements, to place it into kind of a timeline of those large scale imaging projects, because this Human Connectome project is not the first one. The first large scale imaging project was this Alzheimer's initiative APNI study, focusing on one specific neuropathological disease. Then there's a functional connectomes project, but here in 2010 the Human Connectome project started, and the first ended in 2016. And during that time, this kind of a minimal processing pipelines were developed to acquire high quality data and to supply most high resolution processing steps to that time. Just first of all, because those large-scale imaging projects are really cost-intensive, and they're acquiring tons of gigabytes of data, and therefore data must be shared and reused to get the value out of those really large and effortful projects. And therefore I have kind of a quick introduction or a quick glossary for the major terms. And the first one is reproducibility, because open science in this way is really important. And yesterday came across this nice illustration I wanted to share with you in this way, because the definition of those terms, everybody heard of those, is aware of those terms and heard those terms before, but I find it quite cool to define the different parts of reproducibility and yeah, the different shades of definitions for those terms, for example. So there are different definitions of reproducibility. For the first one, it's computational reproducibility. So detailed information has to be provided about code, software, hardware, and how this all was implemented. Then there's a kind of empirical reproducibility. So the detailed information about non-computational scientific experiments and observations must be shared and freely available. And then there is statistical reproducibility. So detailed information must be provided about, for example, the choice of statistical tests, the different model parameters and threshold values. And yeah, so all those steps are needed to use data and to reproduce the results found in one study, for example. And there's a differentiation between different terms, so reproducibility, replication, generalization, and robustness of different processing steps or analysis steps, more broadly said. And maybe if I define it now, you can think about how those different aspects of open science are implemented in this human connectome project dataset, and to what extent it is not implemented. So there's this definition from the Turing-Way community. So reproducible is a result when it has the same analysis steps performed on the same data set, and you consistently produce the same answer. So if I share the data and the code that analyzes my data to you, then you will, if you apply the same analysis, the same code for analysis to the same data set, then you will get the same, exactly the same results. And this can be tested with this human connectome project data, because there everything should be published. So the analysis pipelines are published and the imaging data is published. So one should be able to reproduce the exact findings. Then there's replication. So a result is replicable when the same analysis is performed on a different data set and produces qualitatively similar answers. So if we take a look at another imaging study, for example, which have a different data sets, but the input data is the same. So we can try to take, for example, the data for the 1,000 functional connectomes and apply the processing pipelines from the human connectome project. And we can see if we can replicate the results from another data set. A result is robust when the same data set is subjected to different analysis workflows and to answer the same research question. And as you can imagine, in this case, the results were not robust because the same data set was used with different analyses, and the finding diverged greatly. So this was not a robust result. Combining those terms, for example, if we have replicable and robust finding, then we can say that this is a generalizable result, because we have different data and the same analysis, which leads to the same result, and we have the same data and a different analysis. So the underlying ground truth is probably the same. So this result is generalizable. Okay, any questions so far about this? I think not. And yeah, then we can start with some other brain-related terms. So just to keep everyone on the same track, there's a template brain, which is the standardized stereotaxic coordinate frame for data analysis and report, as I told you in the lecture before. And this is a framework for creation for those neuroimaging databases. And yeah, it helps to calculate a population mean. So the common characteristics of a brain over a huge population of acquired images. Just to visualize this, information gets also lost when registering and averaging towards a template brain, because if you can see on the left side, this is a scan of an individual brain, and this is clearly more the resolution is better than a group average, because averaging signal intensities across many subjects will blur the borders between different brain regions. So, and for the cortical surface, you can also see that the folding pattern is also much more simplified, because in an individual brain, the folding is like a fingerprint unique, and those informations yeah, clearly get lost if you average towards group template atlas. Then another term which is important is the parcellation, which is a subdivision of the brain into consistent parts. Yeah, which we touched in 1 minute last lecture as well. So that we can segment the brain into different compartments to communicate where are we in the brain, to supply or to comparability across individuals and studies, or to reduce the dimensionality of complex and noisy data sets. But if you can imagine, you have if you aggregate data towards areas, so if you parcellate data into distinct brain areas, the dimensionalities get reduced from, for example, 160,000 vertices of the surface towards 300 areas of the cortex. And yeah, there are subject parcellations or group level parcellations or atlases. Some parcellations are plotted here. So the Destrue parcellation is distinguishing between gyri and sulci, so the cortical folding or the topology of the cortex, like the desican, which yeah, segmenting the different lobes and the gyri of the cortex. Then there's, for example, the Jülich brain, which is focused on different cytoarchitectural properties of the brain, so how the cell bodies are organized, or and then there's a multimodal parcellation which we focus on today. And yeah, a combination of group average template and segmentation of that is also often called an atlas. So in a broader term, an atlas is a generalizable brain topology or segmentation. And mostly it's a combination of intensity image, so a group average image, and a segmentation which parcellates the group average and assigns labels to the areas of one brain. You can register then an individual image to the template to propagate those atlas labels to the subject. For example, the Jülich brain is based on histological methodologies and can be applied to each individual subject. But if we register the individual brain, we can see where, or at least in a spatial topology, we can then propagate the information from this group average to the single individual, which doesn't mean that everything is then passed over, but at least the spatial correspondence between the group average data and the individual brain is then set. We can also say that a group atlas is a kind of an average parcellation of a template brain, or a parcellation of an average template brain. As another word is brain ordinate. So it's a brain location that is specified by either a surface vertex, so it's a combination of brain and coordinate, obviously, and yeah, it can be a surface vertex or a volume voxel. And brain ordinates include gray ordinates, so gray matter vertices or voxels, and white ordinates, white matter voxels, so the tracts. And the SIFTI file, this is proposed by the Human Connectome Project, is containing a list of those brain ordinates and can handle those combined surface and volume representations. The cool thing is, if you have kind of a brain ordinate coordinate system, it doesn't matter whether you are investigating a voxel or a surface, you have a common framework to point towards those spatial entities in a coordinate framework. Then, yeah, we are now finally coming to the HCP neuroimaging approach and the modalities. They are currently or perceived as really with high quality processing pipelines based on high quality data of, for example, for the 100,000 young adults, which in this Human Connectome project, the focus really was on good data quality and good processing pipelines. So there were 7 Tesla scans applied for tractography, but also many different modalities like myelin maps, task fMRI, functional connectivity, and many more. And the whole paradigm can be summarized in 7 tenets, which the authors self say it's called HCP style. And yeah, the 7 tenets are that there's a large amount of multi-MRI data needed for high resolution and high data quality. We have to rethink pre-processing steps and pre-processing algorithms. So this was back in 2013. So many things have happened in that time frame. But I think it's still a good tenet to rethink and pre-processing steps. And they introduced this common geometrical framework for volumetric and surface data, the SIFTI format, and a group average of high resolution and quality data can then inform like average common ground of connectivity and architecture of the brain. And with this data, we then can build a multimodal brain parcellation, or at least they said that in 2013. And this was then finally published in 2016, and it is in fact the first or one of the only brain parcellations which are based on multiple modalities of image acquisitions. And the 7th tenet is to share everything. Okay, going into detail. So the multimodal data acquisition to inform brain anatomy and physiology. So they thought about that, and from the beginning said, okay, we have to acquire field maps to help image processing, pre-processing, and we need for sure T1 weighted and T2 weighted images. And the cool thing is that the different contrasts, as we heard last week, the T1 contrast or T2 weighted contrast is showing different tissue types, and the intensities are linked to different tissue types. And we can use the ratio between the T1 weighted and T2 weighted image to measure the myelin content in the cortical area. And we can build geometrical models of brain anatomy, for example, the surface reconstruction and segmentation of subcortical areas. The fMRI modalities are for task-based fMRI or resting state scans, as you can see here. to compute then functional connectivity and topographic organization within the brain areas. Okay, I think I'll skip this. So we need a kind of high resolution because the cortical ribbon is really thin, and voxel sizes of 4 millimeters or something will smush together many things. So we need a minimum 2 millimeter voxel size to at least represent the cortical ribbon in one voxel. Then, yeah, precision of data analysis was one really focus on optimizing the data analysis, the image alignments, and the artifact correction. But I will skip this for now because time is running out again. Yeah, and here is the common coordinate framework with the SIFTI gray ordinate, so the brain ordinates, and it's kind of a way to represent the complex geometry of the cortex in a 2D surface for the cortical surface and the subcortical areas in a volume. And therefore, a SIFTI file format is proposed that lists values for 60,000 surface vertices, so 30K vertices per hemisphere, and over 30K volume voxels, which make up around 100K gray ordinates for gray matter, which can be described in this data format. So yeah, as I told you, there are gray ordinates and white ordinates, and the gray ordinate, based imaging analyses reduce the uncertainty in spatial localization and increases power and statistical power. Yeah, as you can see here, different registrations of data. So here on the left side, we have a volume-based registration, which makes the data more blurry because you can register your data in 3 dimensions. So the possibilities of shifting the data are much more diverse than in a surface-based registration. And there, the borders of one area of the Jülich brain here, for example, are more clearly defined. I won't go into detail in this SIFTI file format, but it's kind of using the nifty2 file format for storing the data and the metadata, which is stored in the header of the nifty file. And so it's using the nifty, which is a standard file format neuroimaging, but is kind of adapting it to separate the data for cortical vertices and subcortical voxels. So yeah, why surface-based analysis at all? We've talked about this like now for this 3rd time. So with reducing dimensionality, we can better align the cortical surfaces. We can increase statistical sensitivity and spatial localization in group studies, and we can decrease the risk of signal contamination. So if we apply smoothing, for example, smoothing in 3 dimensions can lead to a contamination of, for example, white matter voxels lying beneath the gray matter voxels. And this doesn't happen if we have a cortical reconstruction where we have only the surface, and there we can smooth only on the cortical surface, which is all gray matter. And as you can see here, the distances between 2 voxels are in fact farther away than in a surface registration than in a volumetric registration. This is a bit misleading because this is also a cortical surface registration. So now it's hopefully getting a bit clearer. So we have a volumetric scan, and the distance measured in a volume is only this. But if we have a cortical reconstruction, then we have to go along the cortical ribbon, and the distance is even increased. So the smoothing, if you apply smoothing, is not, if you have a kernel that has 40 millimeters or whatever, and then you apply smoothing around here and not across the cortical folding. Okay. Yeah, that is just another slide about registration. And this is then the tenet 6, that we have the opportunity with this high quality data to create accurate maps of brain areas. So we want to have a representative sample of well-aligned subjects. We have boundary evidence from multiple modalities, because with the HCP dataset we acquired a lot of different data modalities, and we can use already existing terminology for those areas. So we have a common framework to communicate those results, and we want to have the possibility to replicate parcellations in individuals. So those are key qualities of cortical parcellations stated by the HCP. And compared to different atlases, those qualities apply. Some of those qualities apply to different atlases, but not all of them. And this is quite cool feature. So on the right side, you see the original Brodmann cytoarchitectonic atlas drawn by hand, and functional connectivity networks, cytoarchitectonics, and cortical topology-based parcellations. But the goal of the multimodal parcellation from the Human Connectivity Project was to reliably identify parcels which are distinct from one another in architecture, function, connectivity, and topographic organization. So we have a multimodal data set to distinguish areal features of the brain. And we want to have a kind of a framework that consistently identifies those area features in individuals. So we improve statistical sensitivity and power because of dimensional reduction. We enhance the signal to noise ratio because we are averaging for an area which is distinct, and we reduce the number of statistical comparisons between like hundreds of thousands of voxels in comparison to 360 brain areas. So the HCP parcellation reflects architectonics, topography, connectivity, and function, and is set as a group average of those HCP subjects, but there's also an algorithm developed out of it for parcellating individual brains. But the original multimodal parcellation is a semi-automated neuroanatomist-driven parcellation. So different modalities were averaged across this population of the Human Connectome Project, or a subpopulation, and then neuroanatomists decided which features were relevant to disentangle areal borders of distinct brain areas in the brain. And this led to an 180 area parcellation of the human cortex. Yeah, this is going way too far into detail for the 3 minutes left. So this is just an overview of the modalities used. So we have myelin maps, cortical thickness measures, task fMRI, and functional connectivity measures. And the areal borders of those of the parcellation were measured with gradients. So where are changes in those areal features, and those gradients as the spatial derivatives of the data were then used to identify areal borders. And as you can see here, we combine the data from different modalities and to extract the gradients where the signal is changing, to delineate certain areas of the human cortex. And this was done for 180 areas and for all those 5 modalities. So it was, yeah, the best fitting modalities were then chosen by the authors to define the areal borders. And this came out with 180 areas for each hemisphere. And this is the cool thing, because we have now parcellation, which is functionally relevant. So we have areas that are functionally distinct, but also are informed by cortical architecture, like the folding structure and the cortical thickness and the myelin, which is the way to get cortical, or kind of a cytoarchitecture, but not with the cell bodies, but with the axons or with the myelin. It's a non-invasive measure of brain architecture. So the cluster parcellation integrates different modalities and therefore is it's functionally but also architecturally legit, so to say. And this can be seen here as well. So this was theory, but this can be also measured with a comparison between different parcellation schemata in this study, and we can test the functional homogeneity within the parcels. And here the number of parcels and the homogeneity is plotted, and we can see that the cluster parcellation is really well placed. The other ones that are even more homogeneous are mostly functional based parcellations with one single modality. And the agreement with the original Brodmann areas, which are based on histology and dissecting like post-mortem brains, there we have also a high agreement with established cytoarchitectural areas. So this is quite a nice compromise between functionally distinct and architecturally distinct areas in this parcellation. And there's all, I think that's it for now, but just 1 minute, that there is the possibility to use this kind of framework for an automated individual subject parcellation. But the classifier from the study was not yet released, but the methodology is communicated in a way that everybody who is kind of at least intermediate expert in machine learning can reproduce the classification of areas based on multiple features to create an individual parcellation based on multimodal areal features. Okay, that was my really quick wrap up of the HTTP pipelines. There was a lot more planned, but basically I am at least managed to get the most important stuff till the end. But yeah, if you don't mind, we can take some time for 1 or 2 questions. But for now, I want to thank you for your attendance and for your patience to go through those really complex things in this quite fast fashion with me. So thank you for